start recording, and we'll hand it off to the first tutor.
All right. Hi, guys. I'm going to start sharing my screen. Do you guys see it? Oops, shouldn't start on the first. All right. Okay, so today I'm going to be talking mainly about alpha helices and sort of compare how they fold and interact with different structural elements in soluble versus membrane proteins. So I think last week you guys covered coiled coils. So if you remember, isolated alpha helices in solution are not very stable because they could just as easily unfold and still fulfill their hydrogen bonding requirements with the solution. The solution can, hydrogen bond with them. And so there's not really much motivation for these alpha helices to be, to keep their conformation as helices. So the simplest way to stabilize these isolated alpha helices is just to pack them together. When there are just two alpha helices that are isolated, this is just done through coiled coils where they're like wound around each other. But when there are more than two alpha helices, or they're just part of more complex proteins, they would rather pack against each other and like remain sort of like straight and rod-like. And a good example of this is what is called a 4-helix bundle, where each helix is in coiled coil, but instead what's different is that they pack against each other rather than like wrap around each other. So the alpha helices that pack against each other like this do so in what is called a ridges into grooves pattern. So every alpha helix has ridges and grooves, as I've denoted in this diagram here. And ridges are spaced 3 to 4 residues apart, which hopefully makes sense because we learned that an alpha helix has 3.6 residues per turn, and there is 1 ridge per turn. And basically to pack together, ridges on one alpha helix can insert into the grooves of another alpha helix. So I thought I would walk you through an example that was gone over during lecture, because I know that when I took this class, everything about ridges and grooves kind of made sense until we started bringing like angles and math into it. So, so looking at A first, let's say you have two helices that both make an angle of 25 degrees between the ridges and the helix axis, which runs straight down as such. And we want to pack them into a ridges into grooves pattern. So first we need to get the helices facing each other. The way they are right now, the ridges and grooves of helix 2 aren't facing the ridges and grooves of helix 1. And so we want to turn one of the helixes over 180 degrees. And it doesn't matter which one you turn over, but in this specific example, helix 2 is flipped 180 degrees. So this is what it looks like after it's flipped. So if helix 2 was at 25 degrees, now it's at negative 25 degrees. And so now we want to align the ridges of helix 2 into the grooves of helix 1. And to do that, we want helix 2 to also be at 25 degrees, like helix 1 is. So to match the 25 degrees, we should rotate helix 2 50 degrees to the right. Hopefully that makes sense, because negative 25 plus 50 would put it at 25. And then part B is basically the exact same thing, except helix 2's the angle that helix 2's ridges make with the axis is negative 45 degrees instead of 25 degrees. So if you flip it 180 again, then it would be at 45 degrees, positive 45 degrees. And then helix 1 is still 25 degrees. So if you want to match the 25 degrees, you have to rotate it 20 degrees to the left. 45 minus 20 is 25. So yeah, hopefully that made sense. If it doesn't, I have additional practice problems that might make it clearer. But yeah, I think it helps to like sort of think through this yourself without looking at like what I have written, and I think it'll become clearer. So far we've been talking about proteins in solution, but what about membrane proteins? So remember that the membrane is a lipid bilayer, and therefore it's very hydrophobic. So it turns out that the hydrogen bonding requirements of a membrane protein can actually be completely satisfied by hydrogen bonding with itself. So yeah, like even though the membrane is made up of, fat, like hydrophobic environment, the membrane protein, just by hydrogen bonding with itself, can satisfy all of its backbone's requirements. And the point is that this can only be done by folding into alpha helices and beta sheets. So this means that membrane spanning alpha helices are pretty stable on their own, but in contrast, isolated alpha helices in soluble proteins or just in soluble environments in general are unstable and need interaction with other protein elements. And then I want to mention that the membrane spanning part of a protein is almost always an alpha helix, but occasionally can be a beta barrel, which is basically just a closed beta sheet. So if you think of a beta sheet as a flat sheet of paper, then a beta barrel would be like if you rolled that flat sheet of paper. So what about the membrane proteins side chains? So membrane proteins typically have hydrophobic side chains all throughout, both on their outer face and inner core. Outer face, by outer face, I mean the side of the membrane protein that interacts with the membrane, like the membrane interface, and by inner face, I mean like the core of the membrane protein. So, and you might be thinking, well, it seems kind of more intuitive for membrane proteins to have hydrophilic, i.e. hydrogen bonding interactions in their core and hydrophobic interactions on their outer surface. And so like, why are polar side chains so rare? But actually it's much better to have hydrophobic interactions in their core as well. And this is because hydrogen bonding requires very specific geometries, while hydrophobic interactions just require the residues to be relatively close to each other. So basically, hydrophobic interactions are much less, they're much less picky about the orientation of the residues. And so the specific geometry requirements also means that polar side chains are more sensitive to mutations, whereas a hydrophobic side chain could be substituted by another hydrophobic side chain, and the protein would generally still maintain its function. But that doesn't mean that polar residues don't exist at all. Sometimes they can be found in a membrane protein, and if they are, it's usually because they carry out a specific function. For example, in bacteriorhodopsin, bacteriorhodopsin is basically a proton pump where there are charged residues along the pump that facilitate the movement of protons. So, and then lastly, I just wanted to summarize some important differences between membrane and soluble proteins. So there's almost always a question on the midterms or problem sets about like where it asks you to predict the what location is the alpha helix in based on its amino acid sequence. Is it in a soluble environment or is it in a membrane environment? And so if you have short stretches of hydrophobic residues that are interrupted by hydrophilic residues, then this likely means that the alpha helix is an amphipathic one, and that is usually found in soluble proteins. But if you have longer stretches of hydrophobic residues, and the rule of thumb is at least 20, then it's probably part of a membrane protein. Also, when it comes to folding, soluble protein folding is mainly driven by the hydrophobic effect. Well, yeah, it's pretty much driven by the hydrophobic effect, which is not the case for membrane proteins, since membrane proteins environments are hydrophobic, and the main mechanism of the hydrophobic effect is that the hydrophobic residues of a protein are being sequestered within the core when there is a hydrophilic environment, and so the hydrophobic effect could not apply in a membrane. And then the membrane protein folding is driven by hydrogen bonding, and with some contributions from van der Waals interactions, but mostly hydrogen bonding. But in soluble proteins, hydrogen bonding is actually weakened because of the competition from water. So yeah, I think this kind of explains why a lot of soluble proteins tend to be globular, and membrane proteins are mostly like rows of alpha helices or beta barrels that can be like isolated if they need to. And then I think I'm running out of time, so I'll just remind you guys that these slides are on Google Drive, and then I have the answers to these practice problems on the next slide after this. So if you want to look over this in your own time, please do so. Yeah, I'll stop sharing here.
See my screen? No? See me? No? Okay, sorry. All right. Hello, everyone. I am Lisa. So today I want to talk a little bit about bacterial rhodopsin. But first, let's start from a review. Can you guys hear me? Yes?
Yes, we can hear you.
Okay, sorry. All right, so let's start from a review of membrane protein. As you know, the principal component of membrane is the phospholipid, which consists of the polar head group and the long nonpolar hydrocarbon chain. So the assembly of the phospholipid bilayer with its hydrophobic interior imposed an immense challenge for the charged or polar molecule to cross the membrane, because in a membrane environment, the polar or charged molecule will essentially lose all the hydrogen bond to water without gaining any compensatory hydrogen bond from the lipid tail. But also it imposed a challenge for the membrane protein. How do they stay stable in the extreme hydrophobic environment? All right, so for a simplest membrane protein, it basically has 3 segments. the transmembrane segments, which mostly consist of almost exclusively hydrophobic amino acid. And the extracellular segment and intracellular segment, they are exposed to water. It's just a, yeah. So from the example of bacterial rhodopsin, hopefully I can give you some perspective of how a integral membrane protein functioning as a conduit that provide a pathway for the charged proton, and more importantly, with its intrinsic light capturing retinol, it enforce this unidirectional proton flow from the cytosol to the extracellular space, which established this proton gradient that we use later for ATP production. All right. So let's take a quick look of bacterial rhodopsin's structure elements first. As you can see, it's an integral membrane protein with 7 transmembrane alpha helices. As Boven talked about, the interaction between the interface is mostly the van der Waals interaction between the hydrophobic residues. Notice it is not what we usually think of the hydrophobic effect in a soluble protein. And it also has polar side chain, which has special functions. They can likely help to construct the correct alignment of the alpha helices because the van der Waals interaction is slippery, they are non-specific. They can also facilitate the transport of proton and interact with retinol. So we'll see that later on. But before I talk about the mechanism of proton transfer, I want to review the pKa, the concept of pKa. One definition I found extremely helpful for me is the pKa is the pH where 50% of molecules are protonated. The conclusion is actually derived from the Henderson-Hasselbalch equation. So this equation actually tells us a lot about the how we can think of pKa, because knowing the pH and pKa, you can actually figure out the ratio of charged and the its neutral, or we can say the ratio of the conjugate base and the acid. So that gives you some sense of the extent of protonation based on the comparison of pKa and the pH of the environment. So you might hear people talking about, oh, the molecule has a higher pKa will tend to be a proton acceptor, whereas the molecule with lower pKa will tend to be a proton donor. And when they say this, they're usually comparing the pKa of this molecule with a default pH value, which is 7. So this is just give you some sense of, without doing a actual calculation of this ratio, you can tell whatever a molecule will be a proton donor or proton acceptor. So for example, the aspartate with its pKa of 4 under a pH of 4 environment will likely to be 50% charged, will be 50% protonated and deprotonated. Whereas a aspartate under the physiological pH of roughly 7 will tend to be a proton donor because pH 7 is higher than its pKa, right? So that's also, sometimes we'll hear it's called aspartic acid because it's a proton donor. Fairly straightforward, right? And another really important aspect of pKa is its digestibility. I would like to think the pH of the environment as a global environment, whereas if you have this whereas if you have this amino acid side chain nearby this molecule of interest, usually it will have some electrostatic interaction that has some influence on the stability of the charge. So for example, if you put a positively charged lysine nearby the negative charged aspartate, the formation of the ionic pair will stabilize the negative charge on aspartate, so make it less likely to be protonated. So then we'll say the pKa of aspartate will be lowered because it's basically saying it will be harder to protonate that aspartate when it can gain the when it can gain the stability from interaction with a positively charged lysine. And similarly, if you put a negative charged glutamate nearby the aspartate, the electrostatic repulsion will decrease the stability of the negative charge on the aspartate. So that results in an increase of pKa of the aspartate. It's basically saying it's more likely to be protonated. All right, so I want to talk a little bit about why we will consider pKa in the context of proton transfer chain. Because as we know, proton transfer is essentially picking up proton and passing it down. That's saying it's a chain, it's basically a chain of proton donor and proton acceptor. So in order to be easily titrated, that requires the molecule's pKa as close as its environment. You can think of histidine, which has a similar pKa as the physiological pH. But so within that chain of residue, the intrinsic pKa of each residue are not necessarily similar to each other or necessarily similar to its environment. So we can infer that the rapid shift of pKa of the residues is essential to construct a sequential proton transfer event. And notice this the adjustability of pKa of the residue is made possible by this heterogeneous matrix of protein environment, which can form various hydrogen bonding, maybe put various charge molecule nearby, or etc. And just give you some perspective, you can see this crucial aspartate in the bacteriorhodopsin proton transfer chain has a largely different pKa value just from a snapshot of the structure. So now we understand the importance of the charged side chain in constructing a proton transfer chain, but is the titrability alone sufficient to establish an active transport channel? So we can look at this example of a proline, which you can see it basically has this large hollow hole in the middle. It has some restriction on the size of the molecule it can pass through, but it's mostly non-specific. And the hole allows the passive diffusion of nutrients and waste product. Yeah. So to understand how does that happen, we can imagine a hypothetical protein, basically has nothing in between, just a chain of water molecules. Let's make a very, very important assumption, which is the aspartate A side has a higher concentration of proton and aspartate B side has a lower concentration of proton. And the proton transfer event starts from the aspartate A pick up a proton. Why does it pick up a proton? Because we can imagine the environment is has higher concentration of proton, thus lower pH, lower than the pK of aspartate, so that the aspartate A pick up the proton. And then it will deliver the proton to its nearby water molecules, and through this extensive hydrogen bonding net, the proton will eventually end up on this originally negative charged aspartate B. Why it is negatively charged, we can also think of, we can also imagine its environment with lowered lowered proton concentration will be higher in pK, higher than the pH of the aspartate B, thus it is negatively charged. So then we can see the entire consequence of this transfer event is that it's a proton basically delivered from its higher concentration side to its lowered concentration side. And it's all start from our assumption of aspartate A has a higher concentration and aspartate B is on the lower concentration side. That's saying if I would like to flip the concentration, the direction of proton flow will also be flipped. And that's not right, because we want to maintain the proton concentration in order to drive the ATP production, right? So that's not right. Then how does the bacterial rhodopsin solve this puzzle? It basically it has this retinol, which is light capturing molecule. It will undergo a conformation change upon excitation of light. Upon the excitation, which imposes the directionality of proton flow. So the key event is that upon its excitation, the all trans conformation will become a 13 cis conformation that's higher in energy, and that conformational change move the retinol closer to aspartate 85 that induce the subsequent transfer of proton from retinol to aspartate 85. So we can put everything together in this diagram, but I think I'm running out of time. Later tutor want me to continue, or because I think student can just basically refers to the diagram and see what's going on there. It's pretty straightforward.
You can take another minute or two if you'd like.
Sure, thank you. All right, so let's put everything together in this photocycle, and we can see in this unexcited ground state, all the retinol is in its all trans conformation. Our lysine 216 is protonated, the aspartate 96 is protonated, and aspartate 85 is deprotonated. So upon the excitation, the retinol will undergo a trans to cis isomerization, basically move the shift base closer to the aspartate 85. And that facilitates the transfer of the proton transfer from shift base to aspartate 85. And then aspartate 85 will donate its proton to this water network, yes. Upon the donation of proton from aspartate 85, this protein will again undergo a conformation change that basically reverse the position of a shift base, move it closer to aspartate 96. In the meantime, the water network delivers the proton from aspartate 85 to extracellular space. And because the shift base is now closer to the aspartate 96, it can readily pick up the proton from aspartate 96 and then undergo a spontaneous relaxation, basically back to the all trans conformation. And also aspartate-96 can pick up a proton from the cytosol and then start the cycle again. Yeah, so that's basically all I want to say today. Thank you.
Okay, so today what I'm hoping to talk to you all about is protein structure evolution. So this will include a little bit of a discussion on how exactly evolution in proteins look like and what we can do to maybe assess evolutionary relationships between proteins. So with that, I think a good starting point for understanding protein evolution is just understanding the relationship between primary structure and the actual physical structure of the protein. And I think at this point in the course, I think all of you are pretty well versed in this, that the primary structure is essentially what determines what the protein will look like through a sequence of many steps involving secondary structure, tertiary structure, and in some cases quaternary structure as well. But just to like wrap all of that together, there's a term that actually describes this process, and that's known as the thermodynamic hypothesis. It basically says that because you have a certain sequence of amino acids, there's going to be some conformation that is most energetically favorable in terms of minimizing free energy. And that's ultimately the conformation that a protein will fall into. And that's sort of the native conformation that exists within cells. In some cases, forming a fibril might be more stabilizing than the actual native structure itself, but you can think of the native structure as a pretty good local optimum, if not the global optimum, in terms of minimizing protein, in terms of minimizing free energy. And so with the primary sequence in mind, maybe if we were trying to at least first take an attempt at understanding the relationship between two proteins, a beginning or starting approach would just be to say, well, let's compare the two sequences and see what percentage of the amino acids are actually similar. And that's a decent initial idea, but I think the example, the globin fold, which was presented in lecture, presents a sort of counterexample for why this might not be the best way to approach understanding evolutionary relationships. And so here you can see 8 different globins that are from pretty diverse species. So the first 3 are all mammalian. Actually, they're all from humans, actually. And then here you even have plants as well as clams, worms, so pretty diverse group of species here. And so notice that even though they've come from pretty different evolutionary lineages, they all have 8 alpha helices. They all generally look the same. And functionally, they all contain this porphyrin ring that binds a heme group and ultimately does its job of binding oxygen, which is what the globin protein is fold, what the globin fold is for. But then they are still able to accomplish all of this despite having very low sequence similarity. So here you can just see some of the sequences of these proteins lined up. You can ignore the yellow highlights for now, but notice that the green highlights are the only columns in which either all the amino acids are the same or almost all of them are the same. So even though they all look very, very similar, they have very few amino acids in common. And so the natural question might be, what kind of explains this phenomenon where you have essentially entire regions where you can kind of have lots of different amino acids, but the end result is a protein that still looks very structurally similar to the ones that you see here. And so one of the reasons for why some amino acids are highly conserved is that they play a very vital role in the function of the protein. And I realize that at this point, I'm probably covering some material that has yet to be introduced in lecture, so consider this as a preview, and don't worry if you don't get it immediately. So in the previous slide, the 2 amino acids that were kind of present in every single row of a given column were phenylalanine and histidine. And the reason for that is because they play very important roles in the active site of this protein. So this porphyrin ring, in order to kind of satisfy the bond requirements of the iron atom that's listed here, or that's shown here in the middle of the ring, you need to form a coordinate covalent bond, and the heteroatom of nitrogen on the histidine ring is a pretty good way to accomplish that. And then the phenylalanine is also necessary. Just I think it plays a structurally important role in terms of maintaining the shape of the active site. So because these 2 amino acids are very critical to the functioning of the active site, it's going to take a very long period of evolutionary time in order for these 2 amino acids to meaningfully mutate, because you can imagine that if there's an organism that had, for example, an alanine instead of the histidine, it wouldn't be able to bind heme, and it wouldn't be able to deliver oxygen to the tissues, and it would die. And then natural selection would pretty much have run its course. And you can also imagine another situation in which, even though the amino acid might not be critical to the function of the active site, it might play a very important role in how the protein comes together, its structure. And here you can connect this to the idea of what you saw about ridges into grooves type packing, in which there are only very specific angles between alpha helices that are allowed in order for a protein to come together. And this also is the globin fold. And so you can imagine that if you have different side chains that maybe don't create ridges or that they don't create grooves, you could either change the angle at which the alpha helices come together or make it so that the helices can't come together at all. And then if you don't have the proper orientation of all the alpha helices, you might not get the active site to come together like this. And this is another example of another reason why some residues might need to be highly conserved. But that's just roughly one half of the equation. There are still pretty long stretches in the sequences that we saw where you could have lots of different amino acids. And one of the reasons for this is kind of requires you to think about, even if you have lots of different amino acid sequences, what are they ultimately forming structurally? So as a thought experiment, you can imagine an n-length amino acid sequence. So there are 20 to the n possible primary sequences because you have 20 different possibilities for each of your n slots. But there are actually relatively fewer actual physical domains that emerge as a result of many different primary sequences. So so you're not expected in this course to know the names of these types of protein domains, but just to give you an example of what they might look like, the main point of this slide is that you can have many different primary sequences, but ultimately when they come together, structurally they look very similar, and many different primary sequences can map onto one structure. So that's why you can have lots of different sequences that might not have exact same sequence identity, but they could still form similar structures, such as a beta barrel, for instance, is something you might have seen, or any of these other domains. And so if we kind of put this all together, what this suggests is that in terms of what residues are allowed to vary evolutionarily in a protein, it's going to be the ones that are not necessarily the ones that are at the active site or are performing critical structural roles. And so there are lots of different areas in which they're allowed to mutate. And so next, I want to introduce this idea that some substitutions are still chemically similar to others. So here is a question where I think you can kind of all participate through the chat. So here I have three amino acids, A, B, and C. I have three peptides. And essentially the question I have for you is, which pair is most similar, A and B or A and C? And of course, the differences are highlighted. And these are the one-letter abbreviations for the amino acids. So just feel free to type into the chat whether you think A and B or A and C are most similar. Okay, looks like 2 votes for A and C. So yes, that is the correct answer. So although A and B have more amino acids in common, if you look at the actual chemical nature of the amino acids that they have in common, there are, or the ones that are different between A and C, a lot of them are very chemically similar. So for example, lysine and arginine, they're both positively charged amino acids. And you could examine each one of these, but essentially what you would arrive at is that these substitutions are chemically similar. And to formalize this, the exact terminology that would be used for that is a conservative substitution, meaning that it's chemically similar, and a non-conservative substitution, meaning it's not chemically similar. So an example of a non-conservative substitution would be if you went from glutamate, which is negatively charged, to alanine, which is nonpolar. That's not conservative. And so kind of the last slide I want to go over, and this will probably be a little bit of an introduction to what Chetan is about to talk about, is we can kind of step away from this chemical intuition of understanding the evolutionary relationship between proteins, because ultimately it's kind of hard to quantify in some sense. Because we know that like glutamate is different from alanine, but how do you quantify the difference between alanine versus valine and alanine versus isoleucine? It becomes a little bit difficult because a lot of these amino acids are very similar. So what we can do is if we have some prior information in terms of we have a set of proteins that we all know kind of perform the same role in terms of they all have the same function, and we know roughly what their evolutionary lineage is. And also if we have a good alignment of those proteins, then what we can do is essentially start asking questions from this aligned block of proteins that if I see maybe a phenylalanine here, how likely am I to see it be replaced by another residue? And that's the concept of the Blossa matrix, which I will hand it over to Chetan to talk about now. So, thank you.
All right. Hey, everyone. So like Pujan said, I'm going to talk about BLOSUM. Looks like you haven't quite gotten to this part in the lecture yet. So hopefully just, listen, and hopefully this will be a nice introduction to make some general concepts make sense. This is definitely something you want to come back to and study multiple times so you really understand the background behind this. But let's dive into this. So as we can talk about, there's a limit to sequence analysis because we're not accounting for the properties, the polarity of amino acids. Is it hydrophobic, hydrophilic? Because if you substitute, let's say, an isoleucine with a leucine, those are both hydrophobic residues and they're still involved in a hydrophobic interaction. In theory, nothing should change in terms of function. That's where we get our different types of substitutions, conservative and non-conservative. And if we want to quantitatively measure that, the study evolution, we'll take a more statistical approach, and we'll look at the frequency at which these side chains are substituted for each other. So we look at different sequences and look at how often we find different substitutions, and that yields us the BLOSUM matrix, which stands for block substitution matrix. This class will mostly be looking at the BLOSUM 62 matrix. And the numbers here, as you all can see, all the amino acids are along each side, so you'll find one, go across and find the other, and that's the score. And these numbers are called substitution scores, S sub I of j. And definition of that is they're related, they don't actually equal, but are related mathematically to the frequency that one amino acid from the left side is replaced with another from this bottom part, relative to the probability that the replacement happens by random chance. So we'll get more into that, but here's definition of the positive and negative scores. So a positive score, it's substituted more often than expected by random chance, and that will be favored by evolution, whereas negative score is the opposite, less often. And we're going to say that's not favored by evolution because it's probably that replacement will have some detrimental effect on the function of the protein and won't be able to function like normal. So how we get to these BLOSUM scores in the BLOSUM matrix is we look at blocks. So this is an example down here. Each row is for the same protein, but in different species. And we'll calculate these scores and frequencies for each block. And we'll do that for multiple blocks and then average those. Two things to note. Sequences that you align here in these blocks shouldn't be very related, because if they're really closely related, they haven't had enough time to evolutionarily diverge. And you'll skew the scores such that you'll say, oh, this has high conservation. But then if you look at the same protein in more distantly related species, you'll find, oh, wait, we're not seeing the same high conservation. What happened? That's because it just hasn't diverged enough. So the cutoff score, there are many types of cutoff scores, but for this class, and for this one, it's 62%, which is where we get BLOSUM 62 from. So if it's more than 62% identical, you'll not use multiple of those proteins. The other thing is to account for amino acid abundance. Every species will have different amounts of each of the 20 amino acids. Some may have more leucine, more phenylalanine, whatever the case may be. And if you have more of a certain amino acid, it would make sense that you'll see that pop up in proteins more often. Therefore, you'll see it more often randomly substituted. So does that make sense? If there's more present, you'll see it pop up more often. That's the main idea. And that concept pops up a lot in quizzes and test questions. So keep note of that. So how do we get the scores? Well, first, we need to calculate something called the substitution likelihood ratio, which is defined down here. Oh, I should get my pointer. Here. So L sub I of j. I and j are the two amino acids you're looking at. And it's defined as the frequency of an IJ substitution within a certain block divided by the frequency of that same substitution in the block, but with all the positions randomly scrambled. And let's look at that in terms of the definition we put down. So related to the frequency that one amino acid is replaced with another, that's our numerator here. Basically, how often are we seeing this in nature being substituted, divided by, or relative to, the probability that it happens randomly by chance. So that's what this denominator is, by random chance. So now the difference between numerator and denominator is basically what are you observing in nature divided by what is theoretically going to happen. So I'm going to walk you through an example problem. This is taken from your textbook, so you can also look there. I pulled it completely from there. So here we're looking at substitution score for phenylalanine and leucine in this block. So first we'll calculate the observed frequency, which is our numerator of the likelihood ratio. So that's calculated by how many times do we see this substitution happen in the same column versus divided by the total number of amino acid pairs in a block. So let's take a look. What do we define as a pair or a substitution? So take where this arrow is pointing, this column. We'll define a substitution, or I like to think of it as in one column, how many pairs can I form? How can I get from one amino acid to the other by drawing an arrow? So we'll do that here. So from this phenylalanine, we can draw 1, and then the second phenylalanine, 2. So in this column, there are 2 pairs for phenylalanine and leucine. So you can always make sure we're not going to double count. So we're not going to go 1, 2, and then go backwards, leucine to phenylalanine, 3, 4. We're only going to go one way because we're looking at pairs. You can always count for these other blocks as well, or other columns, excuse me, but as you can imagine, with a lot of amino acids, that'll get tedious. So what you can do is just multiply the number of one amino acid, so phenylalanine in this case, times the number of other in the same column, and that'll get you the number of pairs. So let's just look at the math, and I think it'll make a little more sense. So first, find how many columns do you have both amino acids present. So we got 3 denoted by the arrows here. And then let's look at the first one here. We have 6 phenylalanines here. So that takes our first position in the pair, multiplied by how many leucines we have, which is 2. 6 times 2, we get 12. So we have 12 possible pairs in this column. We'll do it for the other 2. So 2 here, 12 here again. Add them all up, 26 possible phenylalanine leucine pairs, and that'll be our numerator here. Now denominator, total number of pairs. So let's take one column. There are 8 amino acids in one column. And in a pair, we have 2 amino acids, right? So in one column, the first position will be taken up. There's 8 possibilities. So we have 8. And in the second position, because there's already 1 over here, we have 7 left over. So there's 7. So 8 times 7, we get 56 possible pairings in one column, times in this block, 24 columns, and we'll get 1,344 pairings. Now, we want to divide by 2 because we don't care about order. When we did this previous math up here, 8 times 7, we counted like glycine G to E as one pair, but we also double counted going E to G again. So we want to divide by 2 so we don't include that, and we'll get 672. And then for our observed frequency, you have 26 divided by 672, 0.0387. So we calculated our numerator of the likelihood ratio. Now let's look at the random chance. Okay, 3 minutes. So we're going to calculate that. I like to think about this as if we take all the amino acids here and then we're to place them into a bag, what is the likelihood if we were to draw out one amino acid? It would be a phenylalanine. What's the likelihood? It would be a leucine. So that's what we're calculating here with P sub I and P sub J. Think about it as a bag. What's the likelihood of drawing either of those amino acids? Now, in the end, we're looking at random chance of seeing any of the pairs. So we're looking at what's the random chance of me drawing one amino acid, a phenylalanine, let's say, and the next one after replacement will be the leucine. So that's why we multiply these two together using the multiplication rule, because we want phenylalanine and the leucine. We then multiply that by 2 because this math is treating them as independent events, so the order is mattering, but we don't want that order to matter. So we want to include phenylalanine being drawn first and leucine, and then also leucine drawn first and phenylalanine. So we multiply by 2 to include both of those probabilities. So if we go here, we'll calculate individual probabilities first. Probability of finding a leucine out of this whole block. There's 18 leucines divided by the total number of amino acids in the block, rows, 8 rows by 24 columns. We'll get that number. Same thing for phenylalanine, 25 phenylalanines. And then we'll multiply them together and multiply by 2. We'll get our random chance of substitution, 0.0244. Now, let's look down here first. We'll calculate our likelihood ratio, just plugging into the formula we talked about previously, and we'll get a ratio of 1.59 positive. Now, remember, Blosum matrix is reported in terms of substitution scores, not likelihoods. And we want to convert to that by this equation here. And this equation is how you convert back from score to likelihood. Definitely have these memorized or on your cheat sheet, however tests are working this semester. And make sure you keep track of which is which, because when you take the log of anything, really, you end up having an additive property. So by taking the log of these likelihood ratios, we're now able to add substitution scores together. So if we took the substitution score for one pair and another pair and a third pair, we could add those together. If we were just dealing with likelihoods, because they're probabilities, we'd have to multiply them together. Make sure you know that, keep track, but we can plug in this number into our equation, solve, we get a score of 1.336. So that's going to be probably what you'll be asked usually on a test, but some other notes is that, again, what does the score mean? If it's 5, you'll see a substitution 5 times more often than expected by random chance. Remember, random chance is your denominator in this whole equation. But the scores do reflect the properties, the shapes, the polarities of the amino acid. We can best see that by looking at tryptophan. We can see its self-substitution score. So come here, W, going replaced with another tryptophan, is 11. Very high. It's the highest score in this whole matrix. Why is that? Because tryptophan has a really unique shape. It has a 2-ring structure, as you can see here. And there's no other amino acid that really looks like it. So if it's present in a protein, it's kind of the only one that can do that job. Now, tyrosine and phenylalanine, they do have ring structures that look somewhat similar, and you can see that reflected here. Compared to all the other amino acids here, phenylalanine and tyrosine have higher scores, but compared to the score of 11 for a tryptophan substitution, it is pretty low. So just make sure you can understand, if you calculate a score, how does that relate to amino acid shape and other properties? So finally, what's the end goal of this? We can use those scores, those likelihoods, and then can compare how related whole proteins are to each other. So this is also an example from your book, and a common question will get asked in this class. Is protein 1 here, is it more similar to protein 2, or is it more similar to protein 3? So what you can do is you line these up like we did before, except this is going to be 2 at a time. We'll look at their substitution scores for each substitution. So the AM substitution, VO, NS, et cetera, and we'll do that for both pairs and then add them up. And as you can see here, you can also do it with the likelihood ratios, but you will have to multiply. And we find that when we compare protein 1 and 2, we get a higher substitution score than for protein 1 and 3. And that's just telling us that, okay, we're seeing more often in related proteins that these substitutions are happening. So protein 1 and 2 are more related to each other than protein 1 and 3. Kind of would have spent more time on that slide, but go through my slides if you want. I'll post them, and there's some other helpful notes in there. But yeah, I'll just end this by saying definitely ROSM is something you want to focus on and constantly come back and review and understand the statistical concepts behind the math you're doing. Don't just blindly do the math. That way it'll make a lot more sense as you go through this class. Yeah, that's all I have.